going? So this week we're going to be talking about complex genetics. We're going to be talking about some of the exceptions to where Mendel's laws don't exactly work. So these are things that you can still use Punnett squares for. You can still use that tool. Things like homozygous, heterozygous still apply. But we'll be hitting things like some of the complex stuff that can happen. So the goals for today are to discuss when Mendel doesn't work. So you should be able to name some examples and be able to solve the Punnett squares and the problems that show up when Mendel doesn't exactly work for those problems. So first off, we'll start with some lists and kind of give you some examples. So what is not complex genetics? So we're looking at some simple genetics. Called simple because they're, are Punnett square type things. These are things where there's two traits. There's complete dominance. And the idea is that they are greater than 20 centimorgans apart. Greater than 25 centimorgans apart. That's not centimeters, that's centimorgans. It's a distance, for example, in a gene. So if you have, for example, hair color, it would be at least 25 centimorgans away from the eye color. And those would be genes that are unlinked. So these are simple genetics. There's two traits. There's complete dominance. There's no blending, none of that stuff, and they're at least 25 centimorgans apart. So complex genetics, what are some examples? The most common ones you might have remembered would be sex-linked, meaning that the trait is on a sex chromosome. For example, your X or Y. These are traits that could also be sex-influenced or sex-limited, meaning that, for example, if I had a trait for testicular cancer, it would be sex-limited. It wouldn't matter if I had the trait because I don't have any testes that could get the cancer. Sex-influence might be something that has to do with that if you were XX or XY, you're more likely to get it, but you don't have it. So sex-limited, you can only get the trait if you're a girl or boy. Sex-influence would mean it would be more likely you would get it. For example, breast cancer, you're more likely to get it. It's sex-influenced by the amount of estrogen you have, but you could still get it if you were a guy. Sex-influenced. So sex-linked traits are an example of complex genetics because, for example, instead of just doing traditional Punnett square, you now have to deal with XYs, and XX is another component. In addition to sex-linked traits, there's codominance. So these are getting kind of the multiple allele situation, meaning it's not just black and white. With things like codominance, where you can have two traits shown at the same time. You can have things like incomplete dominance, which is a blending. A blending of traits. This is really common in, for example, flowers. The idea is that if you have an RR, you would be red. A WW would be white, but an RW would be pink. Codominance would mean that the two traits are shown equally. For example, in blood types, A, B, and O. So here's a couple examples to get you started. To those, you also have things like multiple alleles. For example, in some animals, you might have black is dominant to gray, which is dominant to white. White being the most dominant, reverse that. You could have traits where you have multiple things. Black would be dominant to gray to white. So white would be the most recessive. If you had a gray GW, you would be gray. But if you had a BG, you would be black, as in multiple alleles. And lastly, the most complicated situation, it would be linked genes, which I'll also hit on today. These are genes that are less than 25 centimorgans apart. Less than 25 centimorgans apart, meaning that when you do recombination, they are more likely, so going back to meiosis, they are next to each other, so they are likely to be inherited together, would be an example of linked genes. So I'll go ahead and do some practice problems, running through each of these different examples to kind of give you an idea of what those look like. First one I'm going to talk about is sex-linked traits. Those would be examples, things like hemophilia, colorblindness. You might also see this a lot of times, we'll talk about it in Drosophila. This is where they found a lot of these sex-linked traits. When you see it inherited, you'll see that a lot of these will happen more often in the guy. So a more often affected guy or male will suggest that it is a sex-linked trait. So the first place you want to start off with a sex-linked trait is your Punnett square, and it's a girl and a guy. So you set it up just like you would a normal Punnett square. X, we'll put our female on the side, we'll have an XX female, XY. Now you can then put in the rest of the alleles, knowing what your probabilities are going to be. Another special thing about this is you're going to be looking at the differences in female and male. So for example, if we had a female carrier, a carrier meaning she's heterozygous. Another term you might want to look up and see if you know what it means, something called hemizygous, which applies to the guy only because he's XY. It's not heterozygous because he has a Y chromosome, but actually hemizygous. So let's say a woman heterozygous for hemophilia, hemophilia being a recessive trait, and let's say the guy does not have hemophilia, he is normal. So then we can do the cross. We have XH, X little h, big H, and so forth. So we can then mate X to the big H, X to the big H. We have a normal daughter. X to the big H, X to the little h. We have a normal daughter again because she is heterozygous. She has a big H, meaning she is normal. Big H meaning normal. Little h meaning hemophiliac. We can then look at the sons. One normal son. Here's the hemophiliac. He does not have another X chromosome to save him. 1/2 of all the sons will be hemophiliac. So we can do the phenotypic ratios and the genotypic ratios. And you'll see that 100% of the daughters will be normal. 1/2 of the daughters will be carriers. While in the gentlemen, 1/2 will be normal and 1/2 will be hemophilic. So in this, it's important to break it down by both girl and guy for a sex-linked trait because the ratios will be different. We're going to end up looking at a dihybrid cross. We know that big A and big B are separate. They're on the same chromosome. There's 10 centimeters difference, or centimorgans difference. So that's centimorgans. So we know there's a 10% chance that they could cross over, and then they could act like that. So when you're setting up your square, you will know that these ones, big A, little b, for a crossover event to occur, is less likely to occur, but in equal amounts. So 10 centimorgans apart, you would split them in half. Both this one, big A, little b, would happen 0.05% of the chance, because at that other percent of the chance, you would have, so you have big A, little b, little a, big B is going to happen also 0.05. So now you have that happens 10% of the time. You can now then go back and find out how long it's going to take to get little a, little b, big A, big B. So to have it have no recombination, it's going to happen a lot more, 90% of the time, or 0.45 probability with each. Using these tools together, you can find out that it is more likely to have an individual. As such, you would do 0.45 squared to get that. So the idea here that's different is that you're going to be using numbers instead of it always being an equal amount. So instead of each time being 25%, it's now going to be more. And this only happens when you're looking at two traits at once. So this will never happen with a monohybrid case. This can only happen in a dihybrid case. And since it involves math, it would be a very likely problem to occur for something that was a grid-in. Rapid summary you through. There are conditions where Mendel does not apply. And those situations are different for each one. So it's a lot of just practice problems. That would be my biggest tip. A recommendation is to practice. And the things I would practice definitely, things like sex-linked traits, multiple alleles, linked traits, and if you're struggling with the linked traits, make sure you feel comfortable with dihybrid crosses. So this week, my biggest tip for you is to practice the problems. Because when you're able to calculate things like the phenotype, which again is your physical traits, and your genotype is really what you're looking at. You're looking at the power to be able to predict what your offspring are going to look like. So again, if you need any help with these, it's very important you ask. Go to your math instincts. You need to ask. You need to practice. It's not one of those situations you can absorb from the book. Genetics is a lot of practice. So take some time, practice. Hopefully that helped you out a little bit. If it didn't, please let me know so I can make these things better for you. But please, 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 I cannot tell you enough. You need to practice. You need to do the problems. Draw the Punnett squares. Don't take shortcuts. Do it the long way. Get it to the end. All right, so genetics, the more complex side. I look forward to talking to you next week, talking about the central dogma. So take care. See you later.